Hello everyone. I welcome you all here for listening my presentation. My name is Ritakshara Harivedi, studying in 5th semester, and I am going to enlighten you about the Mhc locus. Mhc locus is the topic that I have chosen for my Meet 2. Very first, I will take you towards the agenda of my presentation, which includes an introduction, scientists involved in Mhc study, general organization and inheritance of Mhc, different Mhc classes, different allelic forms of Mhc, Msc molecules and genes, structure, and a few differences between Msc class 1 and class 2 molecules. Before covering any subtopic, I will define what Mhc is. So, Mhc is the abbreviation of major histocompatibility complex, and here you can see the term locus. So, locus is defined as a particular position or place where something occurs or is situated. So, when we talk the term Mhc locus together, that means we'll talk about the location where the Mhc is present and what Mhc is called at that particular place where it is situated. Now, let us come to the introduction part. It tells that MHC has a basic function which helps in recognition of antigens by T cell receptors. MHC was first studied as a genetic complex influencing the transplanted tissue rejection in an organism from other organism of same species. Initially, there were three scientists who named Zickernagel, Doherty, and Benekerev, whose studies showed us that MSc has seminal role in determining adaptive response, and they also showed us that a particular set of MSc molecules expressed by an individual influences the repertoire of antigens to which that individual's Th and Tc cells can respond. Now, let us discuss about how MHC is organized and from where it has inherited. So, MHC is organized in such a way that they are linked tightly in the cluster of genes, and their products play a role in two things. First is intercellular recognition, and second is self and non-self recognition. In mid 1930s, there come another scientist named Peter Gorer and George Snell. Peter Gorer has found four group of genes which were encoding blood cell antigens and designated them as 1 through 4. Later, Gorer and Snell found that the group 2 encoding antigen took part in transplanted tumor and tissue rejection. And Snell called these genes as histocompatibility genes, now known as H2 genes. For that, he also got a Nobel Prize in 1980. So, this was the part where we talked about the organization and inheritance of MHC. Now, I will tell you about the MSc classes. MHC is differentiated into three different classes: class 1, class 2, class 3. MHC is a long stretch of genes present on chromosome number 6 in humans. It is known as HLA complex, and when it is present on chromosome number 17 in mice, it is known as H2 complex. The difference in MHC classes that I will tell you later in this presentation. Now comes the allelic forms of MHC. MHC has basically two allelic forms, but before moving on that, I will tell you what allele is. So, allele is defined as the one of two or more possible forms of a gene that are found at the same place on a chromosome. So, those two forms are polymorphic and polygenic. Polymorphic is defined as when many alleles exist for each gene. Polygenic is defined as many different MHC genes exist. When genes lie close together, that means when they are tightly linked, they generally inherit as a unit from parent. Those linked units are known as haplotype. That means when an individual is born, it inherits one haplotype from each parent. That is, it will inherit one haplotype from mother and one haplotype from father. So here I have showed you an example which describes this. In this, you can see that there is one parent with haplotype small p, and another parent with haplotype small k and small k. And when the progeny is formed, it is two different haplotypes, small b and small k. There are two other examples which will clear your concept about the allelic forms of Msc. Okay. Now let us discuss about the Msc molecules and genes. Molecules of both the classes, that is class 1 and class 2, they are membrane-bound glycoproteins, and they are closely related by both structurally and functionally. They are highly specialized anti-presenting molecules, form stable complexes with peptide ligands and display them on cell for T-cell recognition. So this is the diagram of class 1 MHC molecule. I will explain you the diagram rather than reading out the whole text. So it has two different polypeptide chain, alpha chain and beta 2 microglobulin chain. As you can see, the weight of both the chains is different. Alpha chain is of 45 kilodalton and beta 2 chain is of 12 kilodalton. The weight varies because alpha chain has 3 domains and beta 2 has only one domain. Alpha chain has 3 external domains, alpha 1, alpha 2 and alpha 3. And this molecule has 2 domains. One which is far from the center body is called membrane distal domain, and another one which is near the center body is known as membrane proximal domain. Membrane distal domain has 2 domains, alpha 1 and alpha 2. Membrane proximal domain has also 2 domains, alpha 3 and beta 2. This is also known as Ig fold structure. It has a transmembrane segment and a short cytoplasmic tail. It also has a peptide binding cleft over the top of the molecule. The association of alpha chain and beta chain is important because they are required for class 1 expression on membrane, and the interaction of both the chain is also important during the assembly of the class 1 molecule. And now here's diagram of class 2 MHC molecule. Class 2 MHC molecule, in this diagram it shows that it also has 2 different polypeptide chains, alpha and beta, and their molecular weight is also different. Alpha is of 33 kilodalton and beta is of 22 kilodalton, and each chain contains 2 external domain. Alpha has alpha 1 and alpha 2 and beta has beta 1 and beta 2. Alpha 1 and beta 1 comes under membrane distal domain, which is far from the center body, and alpha 2 and beta 2 comes under membrane proximal domain, that means near the center body. Both the chains associate non-covalently. Okay, so this was all about MHC2 molecule. I've also write about the few differences between class 1 and class 2 molecules regarding composition. MHC1 has three alpha domain that has joined non-covalently, and a beta 2 microglobulin, while MHC2 has two different polypeptide chain encoded by different loci. They are also differentiated in the terms of expression. MHC1 is expressed by a nucleated cell, and MHC2 is expressed by macrophages, dendritic, and B cells. They are also different in structure and function. Msc class 1 it present peptide antigen to TC cells, and Mhc2 it is present possessed antigen to TH cells. So this was all about my presentation. Thank you for giving your precious time to my presentation. Thank you.